So I am Shrija Ghosh and I am a PMRF from IIT Kanpur, and I am taking the problem solving sessions for the NPTEL course Environmental Chemistry and Microbiology. The course instructors for this course are Professor Anjali Pal and Professor Sudha Goel from IIT Kharagpur. So in this session we will be discussing questions from the topic cell chemistry, cell biology and microscopy. So we will start now. So question number 1. Bacterial cells can be stained with dash to reveal the presence of lipid inclusions. Option A, safranin. Option B, methylene blue. Option C, trypan blue. And option D, Sudan dyes. So what will be the answer? Which of the following dyes can be used to reveal the presence of lipid inclusions? So we have to select such a dye which can interact with the lipid bodies. Lipids are basically fatty acid molecules, right? So any dye that can interact with these fats can be used to stain these lipid inclusions. So among the 4 options given, Sudan dyes are the ones that can interact with fats. So the correct answer for this question is Sudan dyes. Sudan dyes have a high affinity to fats, therefore these dyes are used to reveal the presence of lipid inclusions. Other than that, the other 3 dyes, safranin, it's a pink colored dye and it is used to stain, it is used as a counter stain in case of gram staining. I'll be covering that staining procedure in the subsequent slides. Methylene blue is a blue colored dye, it's an alkaline dye and it is also used to stain the cells. Trypan blue, it is actually used to differentiate between live and dead cells. So when the cells start dying, the membrane of the cell starts rupturing. Since the membrane ruptures, when we stain these cells with trypan blue, these dead cells take up those blue colored dye and appears blue under the microscope, whereas the live cells, they don't take up the dye and they appear colorless. So trypan blue is used to differentiate between live and dead cells, especially in case of mammalian cell culture. So you can see the cells under the microscope. The cells that appear colorless are the live cells and the cells that appear blue are the dead cells that have been stained with trypan blue. Okay, and Sudan dyes are the dyes that are used to stain lipid inclusions. Okay. I hope this is clear. Moving to the next question. Cell size and cell number can be measured using option A, hemocytometer, option B, Coulter counter, option C, Petroff-Hauser counting chamber, and option D, none of these. So which of the following device is used to measure both cell size and cell number? So hemocytometer, it is used to measure cell number. Coulter counter, whereas we can use Coulter counter to measure both cell size and cell number. Petroff-Hauser counting chamber, again it is used to count cell number only. So both hemocytometer and Petroff-Hauser counting chamber both work under the same principle and they are used to count the number of cells. But Coulter counter can determine both cell size and cell number. So the correct answer for this question is option B, Coulter counter. So I'll show you how it works. So if you see in the first figure, it is a Coulter counter. So whenever a particle passes through this orifice, a signal is generated due to difference in the current fluctuation, and that's how the device captures a cell and recognizes the cell shape, cell size, and number of signals will determine the number of cells. Okay, one by one each cell will pass through the through this orifice and signal will be generated. So number of signals will be equal to the number of cells, and depending on the size of the cell, a larger cell will generate a larger signal and the smaller cell will generate a smaller signal, and depending on that, cell size and cell number both are determined. Whereas both Petroff-Hauser counting chamber and hemocytometer, which is also known as Neubauer chamber, both are used to only count the number of cells. So in the next figure you can see it's a kind of a slide, and on the slide these kinds of grids are formed. So the only difference between Petroff-Hauser counting chamber and hemocytometer is the volume of the cell solution that it can take up. So the depth of the chamber. So the depth of Petroff-Hauser chamber is 0.02 mm, whereas Neubauer chamber it is 0.1 mm. So hemocytometer is generally used to count the mammalian cells or the larger cells, okay, blood cells or any mammalian cell line. Counting such cells we use hemocytometer, but to count smaller cells like bacterial cells we use Petroff-Hauser counting chamber, which has a smaller depth and it can accommodate less number of cells, so that the counting would be easier. So what we do generally, we generally dilute the cells and then we load the cells through these chambers. If you can see on the slide, there is a slit made, okay. In the center you can see both the sides two grids are made. So on one side you can load one of your cell solution, and once you load the cell culture, then under the microscope you can see these grid lines that are made on the right hand side of the screen, and on these grids, if you zoom and if you focus, you can see cells inside each square, and then you can count the number of. So the next question number 3. Most microbial structures and enzymes are composed of option A lipids, option B proteins, option C is carbohydrates, and option D is lipids and carbohydrates both. So we have to tell which of the following macromolecule is present in the most amount in the microbial structures and enzymes. So we all know enzymes are basically proteins, right? And most microbial structures also have proteins as their major macromolecule component. So proteins form up to 55% of the dry weight of a bacterial cell. The virus coat is also made of proteins that envelop the nucleic acid. I hope now my voice is clear.
Yes, ma'am.
Okay.
Is it better?
So if you see in this table, 96 of the percent dry weight of a cell is composed of macromolecules, and these macromolecules comprises of proteins, polysaccharides, lipids, lipopolysaccharides, DNA and RNA. These are all macromolecules. So, out of all the macromolecules, if you see, proteins constitute the largest amount of the dry weight. So, proteins constitute for more than 55% of the percent dry weight for a cell. Then comes the then comes the lipids actually, which is 9.1%, and RNA is 20.5%. Polysaccharides constitute 5% of the dry weight, lipopolysaccharides constitute 3.4%, and DNA only constitutes for 3.1%. Okay, and apart from that, after 96%, out of that remaining 4%, 3% is constituted by the monomers of these macromolecules like amino acids, sugars, nucleotides, and 1% inorganic ions are present. So, you know, many enzymes have copper ions and iron containing proteins are there. So, first those inorganic ions constitute total 1% of the dry weight of a cell. Okay, so out of 100%, 96% of the dry weight is because of the macromolecules present, and out of all the macromolecules, 55% of the dry weight is because of protein. Okay, this is clear. So,
Yes, ma'am.
so we'll move to the next question now. Which of the following organelles contain DNA, divide and possess some degree of autonomy? So, which of the following organelles contain DNA? They can divide and they possess some degree of autonomy. Option A is Golgi apparatus. Option B is ribosome. Option C is mitochondria, and option D is peroxisomes. So, which of the following organelles contain DNA? So, the answer for this question is option C, mitochondria. So, mitochondria, it's a cellular organelle present in animal cells. So, mitochondria, they have evolved from specialized bacteria. Actually, both mitochondria and chloroplasts. So, chloroplasts are present in plants and mitochondria is present in animal cells. So, they both have evolved from specialized bacteria which survived endocytosis by the other cells that got incorporated into the cytoplasm of the cell. Hence, they are known as semi-autonomous organelles. So, basically, during evolution process, some cells have endocytosed. Endocytosis means they have engulfed the cells inside them, and then after endocytosis, so let's say you have a bigger cell and it has engulfed a bacterial cell, a small bacterial cell. So, that bigger cell has the smaller bacterial cell inside it. Now, that bacterial cell, it has somehow survived inside that bigger cell and it got incorporated into the cytoplasm of the cell, and when that particular bigger cell that started dividing into progeny cells, all the progeny cells contained that particular bacterial cell and that became an organelle. So, this is how evolution of mitochondria took place, and that is why these mitochondria, since you know bacterial cells have DNA and most of the bacterial cells have circular DNA. So, these mitochondria are known to have circular DNA, which is different from the genomic DNA of the cell, and that is why these organelles are called semi-autonomous. So, they are half independent, you can say, okay, because they have their own DNA. So, they are a cell in themselves. Okay, so they have a DNA that has certain genes for its function. So like a bacterial cell, mitochondria also has its own genome. Okay, but this this mitochondrial DNA, it is passed on to the next generation through maternal inheritance. So when so generally what happens during fertilization, a female is responsible for contributing the female is responsible for contributing to the mitochondrial inheritance. So mitochondrial DNA is generally received from the maternal line. Okay, so it's called a maternal inheritance. Okay, and it is nothing but during evolution some cells have endocytosed specialized bacteria that got incorporated into the cytoplasm and became an independent organelle. Okay. So we will move to the next question. So question number 5. Calculate the wavelength of light if diameter of the smallest resolvable object size is 250 nanometer and the specimen is observed with 100x oil immersion lens having numerical aperture of 1.2. Okay, so it's a numerical question. We have the wavelength, we have to calculate the wavelength, we have the resolution given. So the diameter of the smallest resolvable object size, it is basically nothing but the resolution, and the numerical aperture is given. So to solve such questions we have a formula where the distance between two resolvable object is equal to 0.5 lambda by numerical aperture, and lambda is the wavelength of the light. Okay, so let's see how to solve this question. So diameter of the smallest object resolvable is equal to 0.5 lambda. This symbol is called lambda and it denotes the wavelength of the light. Okay, divided by the numerical aperture. So in the question we have been given the diameter of the smallest object resolvable is 250 nanometer. The numerical aperture is 1.2. So 250 nanometer is equal to 0.5 lambda divided by 1.2, which is the numerical aperture. So if you solve, if you have to find the lambda, what we will do, 250 into 1.2 divided by 0.5. If you solve this, the answer would be 600 nanometer. So 600 nanometer is the wavelength of the light required to observe this image. Okay.
0.5, how we calculate, like where it's come 0.5, it's not given in the question.
It's in the formula.
Like you can mention the diameter of this,
It's in the formula
like it's a
only.
formula
Yeah,
0.5.
yeah, 0.5
Okay.
lambda by numerical aperture, that is the formula. Okay. So it comes from the formula only. So 250 into 1.2 divided by 0.5, which is 600 nanometer. It's clear?
Yes.
Okay. So the next question, question number 6. Transmission electron microscope is best to observe option A, a stained bacterial smear, B, unstained live tissue when it is desirable to see some intracellular structure. Option C, intracellular details of a cell that is 1 micrometer long. Option D, a sample that emits light when illuminated with ultraviolet light. So, transmission electron microscopy, it's a type of electron microscope. So, transmission electron microscope, it is used to observe which of the following options? TEM. So, in the name only you can see the word transmission is given. That means it can transmit through the cells. So, by using this type of microscope you can see the internal structure of the cell. So, intracellular details of a cell that is 1 micrometer long. So, that is the correct answer for this question. So, transmission electron microscopes use a beam of electrons and magnets instead of light and lenses. So, in a light microscope you know that the illumination source is light and we see through lenses to magnify the image. So, if you want to see a bacterial cell under a light microscope you will use some powerful lenses that can magnify the size of the bacteria so that you can observe. But in case of electron microscope it uses beams of electron instead of light. So, electrons have a higher wavelength, so you can see better resolutions of you can get a better resolution. So, beams of electron is used instead of light and magnets are used. So, electromagnets are used instead of lenses. So, and by using these two the images are generated. So, two types of electron microscopes are there, transmission electron microscopes and scanning electron microscopes. So, by the name you can see scanning means just scanning through the surface. Okay. So, scanning is just seeing the surface of structure, but transmission, since it is transmitting through the cell, transmission can help you see the inner structure, intracellular structure of the cell. Okay. So, here's an it's a very popular image TEM of a phospholipid bilayer. So, this is how first time we got to know that the plasma membrane is in fact a bilayer. It's a bilayer of phospholipids. So, you can see in this image clearly two different layers of membrane can be visible with the intracellular space between them. So, this was taken using a transmission electron microscope. Okay.
Query, ma'am. Just I want to ask it that the microscope like SAM and transmissions microscope, which kind of sample we have to be verify? Like I'm just asking, is it be possible? Can we see the slurry kind of sample, or can we have to be dry the sample then we can see that?
Yeah,
That
generally
I have confusion.
generally the samples are fixed before you are before you subject them through electron microscope, because in electron microscope beams of electron passes uh, over the cell or through the cells, like through your sample. So, even if you want to see a structure of, let's say, any metal or something, like it's not necessary that SEM and TEM are used in case of biology. You can use it to visualize some nanoparticle. So in such cases also, the samples are first fixed on some surface. So in case of SEM, in case of scanning electron microscope, generally the samples are fixed and coated with some heavy metal like gold or platinum, and then the electrons passes through that heavy metal coating, and then I am not sure exactly how, but then the electron based on the electron density of different parts of that structure, you get the complete, you can visualize the complete structure. In case of transmission electron microscopy also, the sample is fixed on a slide or on some kind of surface, and then that slide is kept under the microscope, and the electrons are allowed to pass through that sample. Okay, is that
Okay,
clear?
ma'am. Yes, ma'am. Ma'am, the sample would be on the dry conditions. Like for example, if we take an example of the nanoparticles like this,
Yes,
that nanoparticle, suppose we have to be dry
yes,
it, then we kept it,
yes,
or like this.
yes, it is
If
in
suppose our sample is in the slurry state, is not possible.
no, so
Slurry state
no,
and
no,
all.
no. If the sample is in slurry state, those are also fixed on the. So when you do the fixing of any kind of sample, it eventually, like you can say, it's in the dry state only, because it's now immobilized over a surface. It is you basically make the sample stationary over some surface, then only you can take the photo. If your sample is in a slurry or it's in a liquid form, the sample will have some kinetic energy and it will keep on moving. So in that case, it's difficult to capture the image of the sample, right? So for that, you have to first fix it over a surface, and then you can take the image of it. Okay, is that clear?
Okay, ma'am. Yes, ma'am.
So moving to the next question. Match the appearance of gram-negative bacteria after the following steps in gram staining: crystal violet, alcohol acetone, and safranin. So we have to tell what will be the color after each step of gram staining. So after addition of crystal violet, what would be the color? It's in the name only. The name of the stain is crystal violet. So what do you think the color would be?
I think purple.
Yes. So after crystal violet staining, the cells become purple, and then once you wash it off with alcohol or acetone, what will happen? Alcohol and acetone can wash off the stain, so the
Colorless.
cells will become colorless. Yes. And then safranin, it has a pink to red color, so it will stain the cells pinkish. So what so the answer for this question would be option number C. Yes, option number C would be the correct answer. So what happens in gram stain? Uh, the cells first, the cells are fixed on a slide. So for doing the gram stain, generally heat fixing the cells is fine. So what we do is we take some bacterial culture, we make a thin layer on the slide, like we just spread the culture solution over the slide, very thin layer of culture solution, and then we just heat fix it. We heat the slide very gently, we move it through the flame and we heat the slide. So the cells are now stuck to the slide, they cannot move, they cannot they cannot move basically. So the cells are now fixed over the slide, and now we will first add crystal violet stain. So once crystal violet is added, all the cells are stained purple in color. Okay, then we do uh, then we fix the crystal violet stain with a mordant called Gram's iodine. So, it is an iodine solution, we add that, and after the iodine solution the crystal violet stain is washed off with alcohol or acetone solution. So, what alcohol does is it can wash off the crystal violet stain. Now, what happens in case of gram positive bacteria? So, in the previous class also I had shown you the structures of gram positive and gram negative bacteria. In the gram positive bacteria, the peptidoglycan layer is very thick, okay, and in the gram negative bacteria there is a very thin peptidoglycan layer, and over that an outer membrane layer is there. Okay, so now when the gram positive bacteria are stained with crystal violet, the crystal violet goes deep inside this thick peptidoglycan layer and the cells appear purple. In case of gram negative bacteria, the crystal violet goes in the peptidoglycan layer, but since the peptidoglycan layer is thin, when we wash it with ethanol or alcohol, the stain gets washed off, whereas in case of gram positive bacteria, it is difficult to wash the stain out of this thick peptidoglycan layer. So, it is deep inside that layer. So, even after washing it with ethanol or alcohol, the the stain does not go off completely, and hence after washing with ethanol or alcohol, when you add saffron in, so the saffron will again stain both the cells, but since already gram positive cells are stained with the purple stain, so over the purple stain that pink stain is not visible clearly, but gram negative stain, after alcohol washing the cells become colorless, so they take up the saffron and they appear pink in color. Okay. So, the gram negative cells, since they have a thin peptidoglycan layer within the in the periplasmic space within its inner and outer lipid membranes, they can take up the crystal violet dye and appear purple. Then the dye and the mordant complex made by iodine, I told you iodine solution is added to make a complex, and it is easily removed from the peptidoglycan layer with alcohol, because the peptidoglycan layer is thin in case of gram negative bacteria. So, it makes the cell wall colorless. The colorless cell wall then takes up the counter stain, that is saffronin, and appears pink to red. So, saffronin is generally pink, dark pink in color. So, they stain the cells that color. So, if you will see, if you have a mixture of gram positive and gram negative bacteria, and if you observe this, if you do the gram staining of the cells and then observe, you can clearly distinguish the purple cells and the pink cells. Okay. So, moving to the next question, question number 8. Which of the following pairs is mismatched? Option A, bacterial identification, which is matched with gram staining. Option B, dark field microscope and light passes through the cell. Option C is three-dimensional images, which is matched with atomic force microscopy. Option D is immersion oil, which is matched with prevents refraction of light and its scattering away from the lens. So, among the 4 options, which is mismatched, we have to tell. So, bacterial identification can be done by gram staining, I have already explained. So, the pink cells will be gram negative and the purple cells will be gram positive. So, you can do the identification whether it is a gram positive cell or a gram negative cell based on that. So, that pair is correct. Three-dimensional images, atomic force microscopy is used to determine the three-dimensional images. So, that is correct. Immersion oil. So, generally when you are doing, when you are visualizing a bacterial cell under a light microscope, for the 100x lens we use immersion oil. Basically what it does, it prevents the refraction of light and its scattering away from the lens. So, 100 at 100 in the 100x lens, when the light passes through that lens, if air is the medium between the lens and the sample, the light can get scattered away. So, to stop that scattering, we use immersion oil, which has a higher refractive index than the air, and so that the light passes through the lens and through the immersion oil to our sample. Okay. So, that option is also that pair is also matched. So, the mismatched pair here is dark field microscopy. So, dark field microscopy, they have said that the light passes through the cell, which is wrong. In the dark field microscopy, your field is dark. Then by the name you can see the field is dark and the cells appear light in color. So, the light does not pass through the cell. Okay. So, the correct answer for this question would be option number B. So, dark field microscopy it uses a special condenser with an opaque disc that blocks the light from entering the objective lens directly. Light reflected by the specimen enters the objective lens and the specimen appears light against a black background. Okay, so the background is black and the specimen, the sample that you are that you want to see, that appears light in color. Is this clear?
Clear.
So, coming to the next question, question number 9. Which of the following is not accomplished by fixing bacterial cells to a slide? First is cell structures are preserved, second is cells are killed, third is cell structures are differentiated, and fourth is cells are attached to the slide. So, I have told you right now we do fixing of the cells to make cells attached to the slide. So, that option is accomplished by fixing the bacteria. We have to answer with what is not accomplished by fixing. So, cell structures are preserved. So, when you fix the cells, the cell structures are preserved because you are not doing anything to the cell wall. Cell wall determines the cell structure and you are just taking the cells and making them stick to the slide. So, the cell structures are very well preserved. Cells are killed. Yes, the cells are killed because you are subjecting it to some chemical or some kind of heat to fix it. So, the cells die when they get stuck to the slide. Cell structures are differentiated. So, this is not accomplished by fixing the bacterial cells because since the cells are dying, the structures are not differentiated. You cannot differentiate between live and dead cells and like that. Okay. So, the answer for this question is cell structures are differentiated. Option number C. Cell fixation attaches the cells to the slide so that they can be used for microscopy or any other analysis. It allows most cell structure to be preserved but does not allow cells to be observed in a living state. Okay. So cell fixation, by fixing the cells, we are making sure that the cells attach to the slide so that they can be used for microscopy and other analysis. So to see the cells under the microscope, we need to fix the cells. So that's why we need to attach the cells to the slide. So that's why fixation is important. And it allows most cell structures to be preserved, but does not allow cells to be observed in the living state. Okay. So moving to the next question, question number 10. Suppose you observe that bacteria are moving in your gram stain, you can conclude that the bacteria are gram negative. Option B is the bacteria have capsules. Option C is you did a flagella staining and not a gram stain. And option D is you did not fix the smear properly. So now we have talked about fixing the cells. So you can see what would be the reason for the bacteria moving in your gram stain. I have told you the first step of gram staining is fixing the cells. So if you don't fix the cells properly, you will probably see them moving around under the microscope. That does not mean that the bacteria are gram negative or positive. That can be only determined by the color of the bacteria that is observed under the microscope after gram staining. Capsules, to visualize capsules, there's other kind of stains that are used, so you cannot say by seeing a moving bacteria in your gram stain whether they have capsules or not. Flagella also, so flagella are the structures that are responsible for the movement of bacteria, but for that the staining procedure is different, and then you can see the flagella in such cases. So in case of gram staining, if your bacteria is moving, doesn't mean your bacteria is flagellated or doesn't mean your bacteria is motile. It just means that you did not fix the cells properly in the smear. Smear means the thin layer of cells that we make over the slide. So next question is question number 11. The ability of lenses to distinguish two points at a specified distance apart is termed option A magnification, option B resolution, option C numerical aperture, and D none of the above. So what will be the term for for the ability of a lens to distinguish between two points? So that the term is resolution. So the correct answer for this question is resolution. So basically resolution means the ability of the lens so that it can distinguish between 2 points. So let's say 2 points are 2 millimeter apart and the resolution of the lens is 3 millimeter, then it cannot distinguish between those 2 points as two distinct points. It cannot see those 2 points as two distinct points, it will see it as one point. If the resolution is, let's say, 1 millimeter, then the lens can see both the points as two distinct points. Okay, so between the 2 points, whatever the distance is, if it is if the lens can see that distance as like the points around that distance as two specific points, that means that particular distance is the resolution of the lens. Okay, the term magnification, as you know, it means magnifying, making something appear bigger, magnifying the size. Okay, So resolution, it is a function of wavelength used and the numerical aperture of the objective lens. It is the ability of the lens to distinguish two points separated by a minimum specific distance. So for example, the resolution of the most common optical microscopes is 0.2 microns. That means if two objects are separated by 0.2 micron distance, it can be distinguished as two objects. Okay. If those two objects are now having a distance of less than 0.2 microns, those two objects will appear as one object under the lens. So that lens will not be able to distinguish between those two objects. Okay, that is the definition of resolution. So it means the minimum distance that a lens can distinguish. Okay. This is clear?
Yes, ma'am.
Okay, so the question number 12. Match the following macromolecules in prokaryotes with their location. So you have four macromolecules on the right hand side: nucleic acid, polysaccharides, lipids and proteins, and you have different locations given on the left hand side: cytoplasm, ribosomes, storage granules and cytoplasmic membranes. So you have to tell which macromolecule is present at which location. So what is present in cytoplasm out of all the four options? Nucleic acid, polysaccharides, lipids and proteins. So which of the following macromolecule is present most abundantly in cytoplasm? So proteins are present in cytoplasm. Okay. Now, which of the macromolecule is present in ribosomes? So ribosomes contain the nucleic acids inside them. Then storage granules. What do storage granules have?
I think polysaccharide.
Yes. So polysaccharides is present in the storage granules, and then cytoplasmic membranes. So membranes are made of, I told you membranes are made of phospholipid bilayer, right? So lipids are present in the cytoplasmic membrane. Okay, so you have cytoplasm with proteins, so 1, 4, then ribosomes with nucleic acid, so 2, 1, storage granules with polysaccharides, so 3, 2, and cytoplasmic membrane with lipids, so 4, 3. So the option number C is the correct answer. So moving to the next question. Ribose and adenine can be found in. Option number A is ATP and RNA. Option number B is proteins and lipid. Option number C is DNA and RNA, and option number D is ATP and DNA. So in which of the following molecules both ribose and adenine are present? So if you see, the full form of ATP is, what's the full form of ATP?
Ma'am, that concept I'm not understand. Like ATP now, that I have to be asked. I want to ask. So you please explain that concept of ATP. I'm not understand in that lecture also.
Yeah, I will explain that. So, yes, in the chat we have an answer. Adenosine triphosphate. So, adenosine or adenine triphosphate, that is the correct answer. So, adenine is the nitrogenous base that is present. So, adenine is present in ATP. So, what is DNA? DNA is deoxyribonucleic acid. So, DNAs have deoxyribose sugar and RNA have ribose sugar. Okay, so the molecules that have both ribose and adenine, that will be option number A, ATP. So ATP has both adenine base and ribose sugar, and RNA also has both ribose sugar and adenine along with other nitrogenous bases. So RNA and DNA both have four nitrogenous bases: adenine, guanine, cytosine, and in place of thymine, uracil is present in RNA. So in DNA, thymine is present. In RNA, uracil is present. Okay, so the correct answer for this question is option number A. So if you see uh, in this slide, you can see the structure on the right hand side. This is the structure for ATP. So it is nothing but it has a adenine nitrogenous base. So you can see that nitrogenous base at the top. Then from that nitrogenous base, the ribose sugar is added. So the difference between ribose and deoxyribose you can see in the figure on the left. On the second position, in the ribose sugar, OH is present, and since it is deoxy means the oxygen is missing. So in the second position, instead of OH, only H will be present in the deoxyribose. So these are sugar molecules. Okay, so ribose sugar is present in ATP with the nitrogenous base, that is A, that is adenine, and then 3 phosphate. So it is adenine triphosphate. Okay, adenosine triphosphate. So difference between adenine and adenosine is basically when the adenine nitrogenous base is connected to the sugar, it is called adenosine triphosphate. So you can see 3 phosphate molecules are connected. Okay, so that is adenosine triphosphate. This is a so ATP is generally associated with energy. So ATP can get hydrolyzed and release one phosphate molecule and then convert into ADP, that is adenosine diphosphate, and that phosphate molecule released is in form of energy. Okay, it gives energy to the cell by getting hydrolyzed. Okay, so that is the concept behind ATP. So you must have studied in school that mitochondria is the powerhouse of the cell. So that mitochondria actually helps in producing these ATP molecules. Okay, is this clear now?
Ma'am, what they reducing?
So this ATP molecule is there, right? So you have 3 phosphate attached to this. You can see the 3 phosphate, right? So from these 3 phosphate, one of the phosphate molecules get removed. So once you remove once you remove one phosphate, it will become diphosphate. Right now it is adenosine triphosphate, so it will become adenosine diphosphate. Okay, sorry. Yeah. So it will become from adenosine triphosphate, it will become adenosine diphosphate. So basically it is getting reduced from ATP to ADP, from triphosphate to diphosphate it is reducing, and in turn it is generating energy. It is giving energy to the cells. Okay. Clear now?
Yes, ma'am.
So we'll move to the next question. Question number 14. True or false? First statement is nucleic acids are found in storage granules. And second statement is proteins are found in cytoplasm, cytoplasmic membrane, cell wall and flagella. So both the options are true, both the options are false. One is true, one is false. You have to answer. So nucleic acids are found in storage granules. Is it true or is it false? Yes, it is
False.
false. So nucleic acids are not found in storage granules. In prokaryotes, it is found in the cytoplasm. In eukaryotic cells, it is found inside a nucleus. So nucleic
That's true.
acids are not found
Answer
in
D.
storage granules. Okay, and the second statement, it is true. Proteins are found in cytoplasm, cytoplasmic membrane, cell wall and flagella. So proteins are the basic building units. So they are found in most of the structures of a cell. Okay, so it is found in cytoplasm, it is found in the cytoplasmic membranes. The membranes have certain proteins that helps in the transport. It is found in the cell wall because cell wall structures also have certain amino acids that makes peptides. Then flagella also have so many structure. Most of the cellular structures are comprised of proteins. Okay, so the second statement is true. So the answer is option number D. First statement is false, second statement is true. So nucleic acids are found in the cytoplasm of the microbes as DNA in the and inside the nucleus of eukaryotes. In case of eukaryotes, it is present inside the nucleus. In case of prokaryotes or microorganisms, it is present in the cytoplasm. In viruses, the instead of DNA, sometimes RNA is present as the genome. So both types of nucleic acids can be present. Storage granules actually contain carbohydrates, and so by name you can see it's storage. Its function is to store. So what do the cells store? They store food. Okay, because the cells need to survive, so they store food. So storage granules contain carbohydrates or other substances that may be needed by an organism when the substrate or the food in the environment is scarce. So basically how we keep some or the other food stored in our houses, our cells also keep some kind of food sources stored inside them, so that if something, let's say there's no food outside available, so immediately you won't die of starvation, right? So if you have some food present at your home, if all the food sources are closed down outside, still you can make something and eat at your home, right? So similarly the cells have storage granules where they store food for emergency purposes. Okay, when the environment is scarce, when the environment doesn't have any food, the cell should not immediately die of starvation, so they can have something already preserved, already stored. Okay, this is clear. Okay, question number 15. Match the following monomers to their polymers. So here we have monomers given: sugars, amino acids, nucleotides, fats or fatty acids, and then polymers are given: nucleic acid, polysaccharides, lipids and proteins. So we have to say which of the monomers join to form the polymers. So sugars, first option is sugars. So what will you get if you attach many sugar molecules together? What will be the polymer called?
Ma'am, sugar is a polysaccharide.
Yeah, sugar is not a polysaccharide. Sugar is the monomer of the polysaccharide. Okay. Sugars are the monomers of polysaccharide. So sugar is one molecule. So many molecules of sugar will get uh, connected to make a polysaccharide. Okay. Similarly, many amino acids will attach to form a protein. Okay. Amino acids get connected by peptide bonds, and many amino acids, when connected through peptide bonds, they make up a protein. Okay. So protein is the polymeric form and it is made of amino acid as monomer units. Okay. Then third is nucleotides. So many molecules of nucleotides will make up nucleic acids. So the DNA and RNA we were discussing. So each unit of these DNA and RNA molecules, these are called nucleotides. Okay. So the nitrogenous base, the sugar backbone and the phosphate group, they all make up one nucleotide. Okay. So ATP is a nucleotide and many ATP, CTP, GTP, they all get connected to make a nucleic acid molecule. Then fats or fatty acids, they make up the lipids. So lipids are made of fats or fatty acid molecules. So many fatty acid molecules connect to form a polymer called lipid. So question number 16. Which of the following is not a function of nucleotides? So you have to say which of the following is not a function of nucleotides. Okay. Then participate in oxidation reduction reactions in the cell, carries sugars in biosynthesis of polysaccharides, regulate, inhibit or stimulate activities of certain enzymes, and option number D is provide structural integrity to the cells. Okay. So which of the following is not a function of nucleotides? So option number D is the correct answer. It provides structural integrity to the cells. That is not the function of nucleotides. Whose function it is to provide structural integrity? What provides structural integrity to the cells? Cell walls. Okay. So cell walls give structural integrity to the cells, and in some cells cellulose and lignin forms the secondary cell wall, which gives cell rigidity and waterproofing. So Nucleotides do not provide structural integrity. They can participate in oxidation reduction reactions in the cell. They carry sugars in biosynthesis of polysaccharides. They regulate, inhibit or stimulate the activities of certain enzymes, but they do not provide structural integrity to cells. Okay. So, moving to the next question. Question number 17. Most abundant substance in a cell is?
Water.
Yes. So, option A is protein, option B is water, option C is sugar, and option D is none of the above. So, the most abundant substance. So, here you have to see in previous question, in one of the previous questions, they had asked most abundant macromolecule. So, that was protein. But most abundant substance, when they are saying the most abundant substance is water. Okay, if they would have asked most abundant macromolecule in a cell, that would be protein. Okay, but water accounts for 70% of the total cell mass, and it is the most abundant substance in the cell. Okay, proteins comprises of the 55% dry weight of the microbial cell. Okay. But when it comes to substance, it is not the most abundant substance. Water is the most abundant substance. So, name the following according to shape and arrangement of bacteria. So, here two shapes are given. You have to tell which shape determines what. Okay. So, first you can see bunch of spherical cells. Yeah, so bunch of spherical cells is present, and the second you can see chain of cylindrical cell is present. So, option number B, Staphylococci and Streptobacilli, that is the correct answer. Let's see different shapes of cells. So, if you see, a cocci is basically a spherical cell. So, spherical cell is called cocci, and or a single cell would be called coccus, and a cylindrical cell is called bacillus, or multiple cells will be called bacilli. So, coccus and bacillus is the singular, cocci and bacilli are plural. So, when two spherical cells are connected together, it is called diplococci, and two cylindrical cells are connected together, so it is called diplobacilli. Okay. Then chain of spherical cell is present, it is called streptococci, and chain of cylindrical cells are present, that is called streptobacilli. Okay. Then you have four spherical cells present in a tetrad arrangement, so that is called tetrad. Tetra means four cells, so four spherical cells arranged in a cubical manner, so it is called tetrad, and two tetrads together form sarcina. Okay, two tetrads together, so 8 spherical cells, they form sarcina, and irregularly, if the spherical cells are present irregularly in a bunch, it's called staphylococci. Okay. And then what is cocobacilli? Basically a spherical cell which two or more spherical cells which together forms a cylindrical cell. So, it is cylindrical and spherical, both, like it appears spherical from the sides and cylindrical from the edges. So, cocobacilli. Okay. So, it is coccus also and bacillus also. So, cocobacillus or cocobacilli. Okay, this is clear. Different shapes of cells. Apart from this, you have comma shaped cells called vibrio, then spiral shaped cells called spirochetes or spirulina, like that. You have different cell structure, like based on the cell shape you have different names of the bacterial cells. Then moving to the question number 19, plasma membrane is considered a barrier to the environment because it is made of structural proteins, it is a rigid structure, no molecules may pass through it, or it controls the passage of the molecules into the cell. So, plasma membrane is considered a barrier to the environment because it controls the passage of the molecules into the cell. So, a plasma membrane, it is called a semi-permeable membrane. What do you mean by semi-permeable membrane? It means permeable means it allows passage of all molecules, so it is permeable. Semi permeable means it can allow the passage of some molecules. So, some molecules it will allow to go and some molecules it will stop. So, it controls the passage of the molecules into the cell.
Ma'am, but plasma we are using for giving the treatment of lots of things in the not only in biological, not only in the biotechnology field. So, ma'am, how it's be barrier? Because I read some papers is there mentions ki plasma like plasma we are using for
That is
what do you say? They can do the both are different, ma'am. Like
yeah,
I'm not
that
understand. That's why I'm
plasma
asking.
and this plasma membrane, it is different. So, that plasma is a part of
Okay.
blood, and this plasma membrane, it's a part of the cellular structure. Okay, so that plasma, it's a part of blood, it contains the antibodies, that's why it's uh, used in different kinds of treatment. But plasma membrane, it's membrane means a covering, okay, covering out of the outside the cell. So, some cells have only membrane, some cells have both membrane and cell wall, okay. So, plasma membrane is that covering of the cell. So, all the cells have plasma membrane. Some cells have a cell wall outside that plasma membrane, some cells don't. And that plasma membrane actually is a very important part of the cell, because without the membrane, without that covering of the cell, the cell will basically, it's like the door of your house, okay. So, if you don't have a door in your house, any person can move inside or outside your house without any regulation, or any insects and all kinds of organisms can enter your house without having any sort of barrier. So, the walls of your house, the windows, the doors of your house, these are basically the covering of your house, right? So, these windows and doors, they are basically selective, so they don't stop you from going out of your house or coming inside, because you you know that, okay, basically your house knows that you are the resident of your house, so you can use that door to come inside and outside of that house. But the things that are not the resident of the house, the door can stop the entry of such things. So, similarly, plasma membrane can actually stop the entry of certain unwanted substances inside the cell. Let's say something harmful to the cell is present in the environment. So, the plasma membrane will protect the cell from that substance, it will prevent that substance from entering the cell. But let's say some nutrition is present outside the cell, so it will allow that nutrition to enter the cell, so that the cells will grow and uh, get healthy, okay. So, plasma membranes, they are called semi-permeable membrane. So they are permeable to certain substances and impermeable to other substances. So the answer for this question is option number D. Yes.
Ma'am, basically that membrane is present in our body or that is present in the plants and all that. Just I want to know that.
Uh, membrane is present outside every cell, even in your body and in the plants or any living organism. Every
Okay.
microorganism also. Every cell has a plasma membrane around the cells. Okay.
Okay.
So the answer is option number D. It controls the passage of the molecules into the cell. Just a second. Can you see my slide?
Yes, ma'am.
So a plasma membrane, it's a semi-permeable membrane made of phospholipids, and these phospholipids have amphithetic properties. So what do you mean by amphithetic properties? It has both hydrophilic and hydrophobic properties in the same part. So if you see in this figure, the black circular part which is present on the outer side, so it faces the water, like water containing environment, fluid environment. Okay, and those parts are hydrophilic in nature, whereas the linear, the straight lines that are facing inwards, those structures are hydrophobic in nature. So plasma membrane, it is made of phospholipids, and these phospholipids have a polar head group which is hydrophilic in nature, and fatty acid, non-polar fatty acids, that is the lipid part of it, which faces inwards, and it is hydrophobic in nature. Since it is hydrophobic in nature, it does not even allow water molecules to enter the cell. So for such movement, there are certain protein molecules present, like that, those are embedded in these membranes, which can selectively allow things that are required by the cell to pass through these membranes. So through these membranes, only gases can pass through diffusion, and no other molecules can pass through the membranes. They can only pass through these specialized proteins that are present, that are embedded in the membrane. Okay. So moving to the next question, question number 20. Active transport is different from facilitated diffusion because active transport requires option A, osmotic pressure, option B, lipid bilayer, option C, energy in the form of ATP, and D is transporter protein in the plasma membrane. So active transport, by the name you can imagine, it's a transport which is driven by some form of energy. So correct option for this question is, correct answer for this question is option number C, energy in the form of ATP. So different molecules move across the membranes of the cell using the cellular energy in the form of ATP. So I have explained how ATP can provide energy to the cells, and using that energy, uh, cells take up certain molecules inside, and that is called active transport. So There are different types of transport: active transport, facilitated transport, or facilitated diffusion. So, facilitated diffusion, by name you can see it is facilitated by something. So, facilitate means it is being it is helped by, like that transport needs help of some other molecule or some other protein. So, the proteins that I have showed present in embedded in the membrane, those proteins actually help the transport of such molecules. So, that is facilitated transport. Whereas, active transport, it requires, along with the proteins, it requires the energy. So, energy is very important in case of active transport. Facilitated transport or facilitated diffusion, the cells do not require energy. The transport occurs through the diffusion process, but using these transport proteins that are present in the membranes. Okay. So, in process of osmosis, what happens? Any molecule that is present in higher concentration on one side and lower concentration on the other side, the molecules will move from the higher concentration to lower concentration. So, let us say if a cell is present and water, the cell is having less amount of water, and it is and if you keep that particular cell inside a bucket of water, the water will go inside the cell and the cell will swell up, because the cell was having less amount of water, and now it can see that the water is present outside. So, it will take up the water inside the cell. So, so that is process of osmosis that occurs. Okay. So, moving to the next question, which of the following linkages are present in cellulose? So, cellulose is a polymer. So, which of the following linkages are present in that polymer? Alpha 1,4 glycosidic bonds between D-glucose molecules, beta 1,4 glycosidic bonds between D-glucose molecules, and alpha 1,6 glycosidic bonds between D-glucose molecules to form branches. So, first of all, cellulose, is it a straight chain or is it a branched molecule? First, we have to see that. So, cellulose is a straight chain molecule, and it has beta 1,4 glycosidic linkage. So, the answer for this question is option B, beta 1,4 glycosidic linkage. Alpha 1,4 glycosidic bonds between D-glucose molecules to form straight chains. It is present in starch or amylose. So, starch molecules have alpha 1,4 glycosidic linkage, and glycogen molecules have both alpha 1,4 and alpha 1,6 glycosidic bonds, and it is a branched molecule. So, it has linear chains, and then linear chains are further branched by alpha 1,6 linkages. Okay. The answer for this question is option number B. The next question, which of the following nitrogenous bases is a purine? So, out of the four options, adenine, thymine, cytosine and uracil, which of the following base is purine? So, the answer for this question is option number A, adenine. So, if you see in this chart, there are 3 pyrimidine bases and 2 purine bases. So, the name pyrimidine and purine comes from the ring that is present in these nitrogenous bases. So, pyrimidine bases have pyrimidine ring present with side chains. So, cytosine, thymine and uracil, these are pyrimidine bases, and purine bases have purine rings present, and adenine and guanine are the purine bases that are present. Okay. So these are the structure of different nitrogenous bases. These nitrogenous bases are connected with the sugar molecules, either ribose or deoxyribose, and then phosphate molecules, and those phosphate molecules are then attached to the next nucleotide, making a phosphodiester bond, and that is how a nucleic acid polymer is made from each nucleotide. So one nucleotide is basically the nitrogenous bases, ribose sugar and ribose or deoxyribose sugar depending on DNA or RNA, and the phosphate moiety. Okay. So moving to the next question, membrane proteins that transport substances from one side to the other are called, so antiporters, symporters, uniporters, or none of these. So what do you call the membrane proteins that transport substances only from one side to the other? Sorry. So option number C, uniporters, is the correct answer. Uniporters, by name you can see uni means one, so it allows the movement of one substance from one side to the other at a time. Symporters means it can allow movement of two substances together in one direction. So symporters means similar or same direction. Antiporters means two different directions. So it can allow movement of one molecule from one side to the other and the other molecule from the other side to the previous one. So membrane proteins that transport substances from one side to the other are called uniporters. So here's an diagram that is showing the different membrane protein transports. So uniporter, symporter, antiporter. So uniporter, you can see movement of one molecule from one side to the other. Symporter, movement of one molecule from one side to the other along with another molecule. So you can see both the molecules are being transported to the same direction. An antiporter, one molecule is transported in one direction and the other molecule is transported in another direction. Okay. This is clear?
Clear.
Okay. So the next question is arrangement of flagella, where tuft of flagella is located at one end of the cell is called. So different arrangement of flagella have different names for these microbial cells or bacterial cells. So bacteria having single flagella have a different name. Bacteria having tuft of flagella located at one end has a different name. So like that. So tuft of flagella located at one end, it is called Lophotrichus. Okay, the answer is option number C, Lophotrichus. So here's a diagram for the flagella. You were asking about this, right? So you can see it's a structure that helps the bacteria to swim or to move. Okay, like birds have wings that help them to fly. Similarly, these bacteria have flagella. These black structures are the flagella that is present. So one flagella present at one end, it is called monotrichous. Okay, monotrichous arrangement. Two flagella present, one at each end, it is called amphitrichous. Tuft of flagella present at one end, it is called Lophotrichus, and flagella all over the body, it is called peritrichous. Okay. This is clear? So you were asking about the flagella, what are flagella? So now can you picture what a flagella is?
It's clear, ma'am.
Yeah. So this was the last question of this session. So if you have any doubt in any of the questions, let me know.
Ma'am, I have doubt. Not the actually, ma'am. This last session is very difficult. I feel it. So I try to grab all the stuff, but not all properly grabbed. So, ma'am, whatever you tell, I understand, ma'am. As exam point of view, ma'am, just can give me some your point of view. Just I'll go with it because some part I'm exactly not understand. Just for example, I'll give example of ATP concept. I'm not understand. Like I'm not pure belongs to chemistry or biological background. That's why
Okay,
a little
that
bit
is,
I feel.
yeah, yeah, I understand. May I know your background? What are you doing now?
Ma'am, belongs to textile background, ma'am.
Okay, so you are a bachelor student?
No, no, ma'am. I'm a scholar, research scholar, PhD
Okay,
student.
PhD student. Okay, so I can understand that it would be difficult for you to imagine these bacterial cell structure and all. So I would suggest you to go through the book that have been recommended in the lecture series. So Tortora, it's a very nice book to understand the microbiology concepts. So you can read, and it's easy to understand also. So you can read that book for clarity. Other than that, like for like taking your example only, ATP, what in particular have you not understood? If you can clarify, then I'll be
Ma'am,
able
like
to
what is
explain.
ATP? Like, uh,
So
yes, ma'am. Like, and what is the role of that in the part of, like, if we talk about ma'am, ma'am is just teach about the ATP. Like which point of view? Like, if you can say that ki it's work on your human body or it's work on the
okay,
plants or
okay,
like.
I'll explain. So these whatever we are studying right now, na, the cell wall or membrane, we are mainly focusing on the microorganisms, the bacterial cells and all. But in general, so if you see in this slide, these five bases you can see, right? Pyrimidine and purine bases. So three pyrimidine bases and two purine bases. So these are the basic structures of five different nitrogenous bases that is present in DNA and RNA. So DNA and RNA are the you can say these are the genome, like they contain the information of all the organisms. So okay, so human body
Okay.
also have a DNA, plants also have a DNA, any animals that you see around yourself, every organism, every living organism has a DNA or a RNA that contains information about every uh, like about that particular organism. Okay, so your DNA contains your information. Okay, so and that information comes from your parents. Okay, so that is how the information is passed on through the generation. Now those information, imagine in terms of codes. Okay, so
Yes, ma'am.
so those informations are stored in terms of codes, like you have certain computer programs, algorithms are there, right? So like that, certain in these informations are encoded in these DNA molecules or RNA molecules. And these RNA and DNA molecules, they have these nitrogenous bases continuously attached. Okay, so if you see a DNA sequence, it will have like an adenine base, then a guanine base, then let's say a cytosine, then thymine, then again adenine, like that. So every person will have a different sequence. So let's say my DNA is some ATGC, GCA, like that, and yours is like ATG. CCAG, like that, something like different sequence. Okay, so that sequence actually differentiates different organisms. So you are different from your parents, or your friends, or you are different from, let's say, any animal or a plant. Why are you so different from a plant? Because both of the organisms, both of like you and the plant, both of you have a DNA, but that sequence of DNA, the size of DNA is very different. Okay, now in that DNA, this adenine nitrogenous base is present. So these nitrogenous bases, you can see, these are connected to a sugar. So I've shown you the ribose and the deoxyribose, right? I'll go to that slide. So here you see, this is the structure of ATP. So the adenine nitrogenous base that you saw in the previous slide, you can see from this particular nitrogen a ribose sugar is added, and from the fifth carbon of the ribose sugar, a 3 phosphate groups are added. So this is a ATP molecule, and this is called a nucleotide. So similarly you will have GTP molecule or CTP molecule, where instead of adenine, guanine will be present, or cytosine will be present, or thymine will be present. Okay, so those nucleotides, they connect to each other to form a DNA or a RNA molecule. This is clear. So from these nucleic acids, this ATP molecule has other advantages other than making up the DNA or the RNA molecule. Now this ATP, it has the capability of getting hydrolyzed, getting reduced into ADP. So it gets hydrolyzed, one of the phosphate group is released, and that release of phosphate group happens with the release of energy. That energy is taken up by the cells. So all the cells that are present inside you, so any work that you are doing requires energy, right? So if you are walking, if you are running, if you are studying, whatever you are doing, even when you are sleeping, your heart is working. Okay, so your heart is beating, so it requires energy for doing such functions. So that, from where will you get that energy? That energy comes from these ATP molecules. So the food you eat, when that food gets digested and all the cellular metabolism steps takes place. So let's say you are eating something, let's say you are eating a chapati, so it's carbohydrate basically. So carbohydrates basically have sugars. So the basic molecule of carbohydrates will be glucose. After digestion you will have glucose. That glucose will be processed in your cells, and during that process certain molecules of ATP will be released. Those ATP, the released ATP, those will be used up by your cells to perform, to function. Okay, so for doing any function, for even for your heart to beat, it requires energy. Okay, it's also work, right? Heart beating, it's a work of the heart to beat. So for such things, so in plants also, different cells of the plants have different metabolic processes. So during photosynthesis energy is formed, so that energy plants uses to grow, to survive basically. Okay, so that is how these molecules are helpful. So similarly when you are breathing, So when you are breathing, that whole respiration process also uh, provides energy to your cells. So it's a give and take process. So you are breathing and you are giving energy to your cells, and when you are breathing, your lungs require energy for doing the breathing process. So the body is getting energy and making energy at the same time. Okay, so for that purpose this ATP molecule is important. Is that clear? Did you understand the basic concept of
Yeah.
ATP, like what is it
Yes.
doing? It's a chemical molecule only, but then it is providing energy to all the cells. Like when we say cells, it doesn't mean any particular organism. Cells are present in all the living organisms, plants,
Yeah.
bacteria, humans, dogs, cats, whatever animals you are seeing around yourself, whatever living, anything that you consider living has a cell, and whatever has a cell will have a cell membrane, a cytoplasm, will have proteins, will have DNA or RNA, everything. Okay, is it clear now?
Clear, ma'am. Ma'am, I have another question not related to this chapter. It's quite related to the first chapter, like the molarity. So, can I ask, ma'am,
Yes,
please? Yeah, ma'am. Actually, like in that chapter, we studied the moles and how to calculate and all. I have one like this. I'll try the questions for exam and practice. So I have one question. I'll just say, ma'am,
yeah.
1 minute, just to give a. Ma'am, how can we calculate? Suppose if we have one polymer, like give take one polymer, like an example of polymer is the chitosan, for example. Suppose we have, ma'am, chitosan 97.5% BDA, and uh, hello, ma'am, are
Yeah,
you
yeah.
listening? Hello,
Yes, hello. Hello.
ma'am, am I audible? Ma'am,
Yes, you
suppose
are.
we have a, 97